This poster outlines the paper named Sensory Axons Induce Epithelial Lipid Microdomain Remodeling and Determine the Distribution of Junctions in the Epidermis by Rosa, Nossman, and Sagasti in 2022. Specifically, my poster explores the experimental question: what is the effect of sensory axons on the organization of basal keratinocytes in the zebrafish epidermis? This sets us up to dive into the intricate relationship between sensory axon and basal keratinocytes in the zebrafish epidermis. The dynamic interplay between neuronal and epithelial components in tissue organization and function will be discussed. Previous studies have shown that epithelial tissues are intertwined with their network of sensory neurons. The interaction between the two play crucial roles in sensory perception, wound healing, and tissue homeostasis. The focus here, expanding on this past research, is to understand how sensory axons influence the organization and behavior of basal keratinocytes, which are the primary epithelial cells in the epidermis. The research methodology utilized live imaging techniques and membrane reporters in order to visualize the interaction between sensory axons and basal keratinocytes. These and other approaches allowed the researchers to observe real-time changes in cellular structures and dynamics within the zebrafish epidermis. Building upon previous research observations, the researchers hypothesized that the presence of sensory axons would significantly impact the organization and function of basal keratinocytes. The expectation was that sensory axons would actively modulate cellular processes within the epidermis, influencing lipid remodeling, actin dynamics, and junctional complexes in basal keratinocytes. The findings from the research revealed evidence that supports the active involvement of sensory axons in remodeling key cellular components within basal keratinocytes. Specifically, sensory axons were found to influence lipid composition, actin cytoskeleton organization, and junctional integrity. This emphasizes the impact of neuronal signaling on epithelial cell behavior. However strong these findings may be, it is also extremely important to acknowledge the limitations present. The research primarily focused on the epidermis during the developmental stages of zebrafish. Therefore, extrapolating these findings to the mature organism requires caution because tissue dynamics and interactions could differ in future stages of the zebrafish life cycle. Because of this, further studies should be done to explore and elaborate on the epidermis during these adult stages. Looking into the future, researchers could potentially investigate the long-term effects of sensory axon-mediated modulation of basal keratinocytes. Understanding the possible long-term effects from these interactions on tissue regeneration and other processes could offer valuable insights into strategies for implementation of tissue repair and regeneration, particularly within the medical community. The first figure, shown on the bottom left of the poster, explores the question: what is the effect of somatosensory axons on the accumulation of labeled membrane microdomains in basal epithelial cells? From thorough analysis of the figure and its implications, it can be determined that the presence of somatosensory axons increases the accumulation of membrane microdomains. The second figure, shown in the middle of the poster, probes the question: how does the expression of these two different proteins affect the relative enrichment of membrane microdomains? This figure proves that the first protein named was more enriched in microdomains than the second. The third figure, seen on the right-hand side of the poster, asks the question: what is the effect of developmental time on the relative enriched of liquid-ordered and liquid-disordered membrane reporters in actin-associated domains of basal cells? After exploration, it can be seen that actin-associated domain enrichment is greater in liquid-ordered membrane reporters when compared to those in liquid-disordered membrane reporters.